Hello everybody and welcome back to another video. In this concise video, we're going to touch on simple diffusion, facilitated diffusion, osmosis, active transport, and endo/exocytosis. It's going to be a concise video, but it's densely packed with high yield material for biology, biochemistry, and on the MCAT. Before we get started, I want to announce that we just hit 500 subscribers. I really appreciate you all for supporting me and subscribing and watching my videos, but there are some of you who watch the videos and haven't yet subscribed. So if you haven't yet subscribed, be sure to subscribe to stay up to date with all of my content. Also, share these videos with your friends. It really helps the channel grow and I would really appreciate it. All right, let's get started over here. So in order to understand why we have all of these different types of transport when it comes to the plasma membrane, we need to understand something about the structure of the plasma membrane. Now, the other name for it is the phospholipid bilayer, and that kind of gives it away. So you have phospholipids and they're forming a bilayer, two layers. Now, let's zoom in on one of these phospholipids and see what it's composed of. So the structure centers around this glycerol, highlighted in faint green over here, and the glycerol is attached to three different molecules. So we have two fatty acids, which are hydrophobic, and then you have a phosphate group, which is hydrophilic because of that negative charge. So when you throw a bunch of these phospholipids in an aqueous environment, they tend to spontaneously arrange in this format. Why is that? Because the hydrophilic heads are going to interact with the aqueous environment, and the hydrophobic tails are going to cluster with one another in the interior of the phospholipid bilayer. Because of this unique arrangement, only a specific set of molecules can freely move or freely diffuse across the membrane. So that's a good segue into simple diffusion. These molecules include small and nonpolar molecules. So for example, O2 and CO2 are small and nonpolar. They have no problem squeezing in between the hydrophilic heads and then interacting and traversing through the hydrophilic tails and getting to the other side of the plasma membrane. Now, when it comes to steroids or fat-soluble vitamins, although they're not as tiny as oxygen or carbon dioxide, they can still traverse via simple diffusion, traverse the plasma membrane, and that's because they're hydrophobic or nonpolar. Now, when it comes to polar molecules, such as glucose or amino acids, or even charged ions, such as sodium, potassium, or other ions that contain negative charges, they cannot freely traverse the plasma membrane, and that's because they can't interact with the hydrophobic tails over here. So they're going to need facilitated diffusion, which is what it sounds like. It's diffusion that's facilitated by something else. What is it facilitated by? Let's take a look. So the process of diffusion over here is facilitated by way of proteins. We have two types of proteins that can facilitate this process: channel proteins and carrier proteins. Let's start with the channel proteins. So for a lot of the ions, here we have potassium, they transport across membranes via these channel proteins. So essentially it's just forming a way for these potassium ions to bypass the hydrophobic tails and enter into the cell, or out of the cell, of course. Next, let's talk about carrier proteins. They're a bit more complex. So over here, as you can see, they're not traversing the entire membrane. What's really happening is that they'll interact with the outer surface of the cell, and once a glucose molecule, these are glucose molecules over here, once it binds to it, it will then act like a revolving door and essentially flip to the other side of the membrane and allow glucose to enter the cell like such. So you can see what I just explained right here. A conformational change occurs and allows glucose to go into the cell. Now let's consider the factors that will increase the likelihood of both simple and facilitated diffusion to occur. So first of all, we need to understand that both simple and facilitated diffusion are a process by which molecules are going from the area of higher concentration to the area of lower concentration. This is energetically favorable. Think of it as like a ball rolling downhill. So if you put a ball on top of a hill, you don't need to apply energy. Just give it that first kick and then it'll roll down the hill. Whereas if you want to push it up, that's going to require energy, but we'll talk about that when we get to active transport. So with that in mind, let's think about factors that will increase the likelihood of both simple and facilitated diffusion to occur. So one thing that comes to mind is if I have a higher concentration of, say, glucose out of the cell, or in the case of simple diffusion like oxygen, then that will increase the rate of diffusion. Also, if you increase the temperature, then that will also increase the rate of diffusion, and that's simply because when you increase the temperature of something, its individual components start to move faster. So those are the ones that you'll see most commonly on the MCAT. Another thing that increases the rate is increasing surface area, just because you have more area for the molecules to diffuse. All right, so now let's move on to osmosis. Osmosis is pretty straightforward. It's basically simple diffusion applied to water. And by the way, we're keeping track of everything over here, just so you know. So when it comes to osmosis, water will want to go to where there is less water. What do I mean by this? So say we have a cell that contains 2 molar concentration of NaCl, and then it's put in a container that contains a 10 molar concentration of NaCl. Which compartment has less water? Well, of course, the container has relatively less water than the cell, and that's because the container has a higher concentration of solutes. All right, but a better way to think of it is just think of it as the solutes will suck the water. So wherever there is higher concentration, water will go. Higher concentration of solutes, that is. So in this case, water will exit the cell. And typically in your biology class or on the MCAT, you'll see 3 scenarios that will present, so let's go over them. So the first scenario is the one we just discussed, and by the way, we call this type of solution a hypertonic solution relative to our cell. So recall our cell had a concentration of 2 molar of NaCl, outside 10 molar. Because you have higher concentration outside, we call it a hypertonic solution. Water will exit the cell, and you'll see that the cell will start to shrink and shrivel. The next scenario is where you have an isotonic solution. So the outside environment is equal to the concentration inside of the cell. Now, importantly, it's not that you don't get any water movement at all, but it's that you're at equilibrium. So the amount of water that enters the cell will also exit. The same amount will exit the cell. So sometimes they'll try to trick you on the MCAT and they'll tell you that there's no water movement. It's really that there's no net flow of water. Finally, we have a hypotonic solution where the outside or the surrounding environment has a smaller concentration than inside the cell. And what's going to happen here is water is going to flow into the cell, and the cell is going to swell and it can eventually burst. Now notice how I have this O in red over here. That's to remember that hypotonic solutions will make the cell make a big O. So now let's move on and talk about active transport. So here we have a classic example of active transport, and that is the sodium-potassium pump. So imagine these red dots over here are potassium and the blue ones are sodium. Notice how outside of the cell we have more sodium, inside the cell we have more potassium. Now, in order to maintain the cell's membrane potential, we need to maintain a higher concentration of sodium outside of the cell and a higher potassium concentration inside the cell. Now, of course, because of the idea of simple diffusion, or facilitated diffusion in this case, you'll see that potassium will tend to leak out of the cell down its concentration gradient, and sodium will tend to leak into the cell down its concentration gradient. So in order to maintain the concentrations, the cell came up with this clever solution where it uses ATP, it uses energy in the form of ATP to go against the gradient. So it's as if now the cell is pushing the ball uphill. So it pumps potassium into the cell, so from its lower concentration to its higher concentration, and then it pumps sodium outside of the cell. Now, of course, every time it does this, it needs to use ATP as its form of energy. Now, this specifically is an example of primary active transport, and that's because it directly relies on ATP to carry out the process. Now, of course, if there's a primary active transport, then there's probably a secondary active transport, so let's go over that. A classic example of secondary active transport is the flow of glucose into the cell when you have higher concentrations of glucose inside of the cell than outside. So naturally, by the process of facilitated diffusion, you should expect glucose to exit the cell, right? Because it wants to go down its concentration gradient. But we want it to go inside the cell. Unfortunately, we don't have a pump that's going to rely on ATP to pump it into the cell. So what we're going to do is we're going to harness the energy that we previously stored in the form of a sodium gradient. So since we have higher concentrations of sodium over here, there's energy stored within this gradient. So what you can do is you can couple the flow of glucose with sodium. Notice this is different than the sodium-potassium pump. In the sodium-potassium pump, we use ATP to pump sodium out of the cell and potassium into the cell. In this form of flow, we don't use ATP. We use the energy that's in the sodium gradient, stored in the sodium concentration gradient, to pump glucose into the cell. So essentially, you can think of it like this, like glucose is having a hard time entering the cell because of the high concentration. Glucose comes speeding through and says, I'll push you with me, and it goes in, all right? So that's how you can think about it. All right, let's move on to endo/exocytosis. As you can see right here, this is an example of exocytosis at the synaptic cleft. Essentially, you form a vesicle and allow certain material to exit out of the cell. In this case, you have neurotransmitters exiting. On the flip side, you have endocytosis, which is the reverse of exocytosis. In this process, you take up material from outside of the cell into the cell. You'll see two different flavors of it. So you have phagocytosis, or cell eating, where the cell will engulf material from the outside, such as macrophages will engulf bacteria, for example, or other toxins. And then you have pinocytosis, or cell drinking, where the cell will drink material, drink fluid from outside containing dissolved solutes. All right, if you found this video helpful, be sure to like and subscribe to stay up to date with all of my content. If you have any questions, drop them in the comments, and also share these videos with your friends. It really helps the channel grow, and I would really, really appreciate it. Love you all, and I'll see you in the next one.